So I'm going to tell you very briefly about some of the research we do in the lab, looking at modeling metabolic networks. And actually, this is mostly in microbes, but we started doing human modeling as well. What you see on the left is a cartoon of a bacterium with its metabolic network. Real metabolic networks have or the order of 1,500 reactions or so. And we try to understand how microbes use the resources and use byproducts while producing their own biomass to grow. What you see on the top right here is a graph of the dynamic flux balance modeling approach we use to predict the time course of growth of bacteria in green, depletion of nutrients in the environment, and what is most interesting, the production of byproducts such as this red molecule could be acetate that could enable the growth of a new bacteria, the yellow one, giving rise on the bottom to interactions. So we can predict interaction between different microbes based on these mechanistic models. And one of the things we're doing is try to match these interactions that are predicted mechanistically with co-occurrence networks similar to the ones that Evan was mentioning. This is work done by Dilip Kishore in collaboration with a bunch of other people listed below. We're applying this, by the way, also to the study of microbiome in ultracentenarians in collaboration with Tom Perls and others. Next slide. One of the things we're doing in the wet lab now is try to test some of these predictions. This is work done by Alan Pacheco in the lab, taking combinatorial communities in the lab, measuring how they grow on different combination of molecules in 96 well plates and measuring how the abundances of different species after a time course. And this gives us the opportunity, for example, to try and understand how microbial community structure and diversity scales as a function of environmental complexity. And this is going to be very important for understanding how microbial communities grow in complex environments like the gut or other human-associated environments. And one of the things that is surprising is that even if you increase the number of carbon sources seen here on the bottom, microbial diversity doesn't increase as much as one would have expected. So this means there are other factors that influence this diversity. Next slide. One of the things that is exciting about these models is that you can extend them to looking at spatio-temporal dynamics, what we call comets or computational microbial ecosystem in time and space. This allows us to model diffusion and expansion of biomass cells on a plate, giving rise to these different colony shapes. And we're extending this to a number of different systems, also in collaboration with the Korolev and Bhatnagar lab. And we feel that this is going to be very important for modeling complex microbiomes as well as human tissue. Next slide. One of the interesting things that is happening now, the human metabolism model is available, shown this as a matrix. This is the whole of human metabolism represented in a single matrix of all the stoichiometric coefficients of the reactions. And we're starting to do spatio-temporal models of cancer organoids using comets. And the idea is to next study interactions between regular cancer and regular tissue and cancer tissue and start looking at interactions with the microbes. The other aspect for which I think is going to be very exciting to extend our models to studying cancer, these are seed collaborations with the Emily lab and David Scher's lab and others. We start looking at how human regulation. So that's another level at which the model will help.